Welcome to Teach Me GCSE Biology,
a revision podcast for GCSE biology students in partnership with Cardinal Newman Catholic School.
I'm Ria Corbett.
And I'm Sarah Matthews.
Hi, I'm Ria Corbett. I'm a science teacher with biology specialism, and on Teach Me GCSE Biology I am teaching my co-host and little sister,
Sarah Matthews,
GCSE Biology topic by topic to hopefully provide you, our lovely listeners, with an audio revision resource.
Yes, we hope that you incorporate us into your revision and as a part of your learning journey. Dip in and out, listen to the episodes on your weaker topics, or use us as your audio biology Bible. Whatever you need us for, we're here.
Okay, episode 3, Culturing Micro-Organisms. Now this topic is for GCSE biology only. So if you're doing combined science, then this is not a topic that you need to look at, revise or learn. It's not going to be something you're examined on. So this is for people that are doing GCSE biology only. So we're going to look at how bacterial cells divide, just very quickly, talk about binary fission, how bacteria are grown, so how you grow bacteria in a completely sterile way in order to investigate things about them, or use them to investigate different things like different detergents or different antibacterials, that sort of thing. And we're going to describe how to prepare an uncontaminated culture using what we call aseptic technique. Now you'll remember using aseptic technique at school, won't you, Sarah?
Can't say I do.
When you did your BTEC, remember we poured plates?
We did
Remember we did loads and loads of agar plates. We made our own.
Yeah, vaguely.
We made the agar, we sterilised it, we poured them, we grew bacteria.
Wear gloves and change gloves, and we had to open things with our little fingers.
Yeah, so loads and loads of that, basically. That's what we're talking about today.
Okay, I
All right?
vaguely remember that, yeah.
Yeah. So just a quick reminder of what bacterial cells look like. Do you remember anything about that from a few weeks ago?
Yeah, they've got flagella.
Okay,
They've got a cell wall.
Well done.
They don't have a nucleus.
No nucleus, that is a key fact.
But they do have a DNA squiggle in there.
So no nucleus, but they're genetic materials just in the cytoplasm. They have flagellum. What's that for?
To help them move.
So to transport them around.
One other thing, last thing, is they are quite sticky on the outside.
They've got a slime capsule, which allows them to stick to other cells and protection from things like antibiotics, that sort of thing. Good. So cell membrane, cell wall, and then don't forget the plasmids as well. Yeah. Okay. Bacterial cells, they replicate by a process that's quite similar to mitosis, which we're doing next episode. It basically involves one cell dividing to become two cells. But of course, there's no nucleus and things like that. So binary fission, which is the process by which bacterial cells divide, involves them replicating their chromosome, their DNA chromosome that they've got in their cytoplasm, replicating the plasmids, maybe making a little bit more cytoplasm, and then the cell splits in two. So the reason it's not mitosis is because there is no, when we look at mitosis, we can say that again, there's no dividing of chromosomes, like splitting of any chromosomes and pulling them to opposite poles like there is in mitosis. That's why it's not that. So we just copy the chromosome, then we've got two chromosomes, one to put in each new cell. The cell divides into two, so the cytoplasm starts to cleave and pinch, and the cell membrane cleaves and pinches, and eventually the whole thing will just split into two. And that's how they do it. And they can do that as quickly as every 20 minutes. After 4 hours, you can imagine how many cells you could have just from one individual bacterial cell. You should be able to find a diagram of that in whatever revision guide you've got, or textbook or something like that, whatever you're using. Okay, so binary fission, that is how bacterial cells divide. Let's start looking at the process of how we can start to grow bacteria to use it for investigations. So this is my absolute favourite. It's one of my absolute favourites. I love genetics, I love microorganisms and microbiology. So growing bacteria is my favourite. We tend to do this in school, and this is typically how we would do things like this in school. And it's one of the required practicals. So it's one of the practicals that you need to have carried out, at the very least seen done as a demonstration. You will be asked questions directly about this practical. Okay?
Yeah.
We do it to investigate microorganisms, and those microorganisms will be bacteria, and they'll be non-pathogenic. So the ones you use in school are what we call non-pathogenic, which means that they do not cause disease. And we grow them in dishes called Petri dishes. Petri dishes are just small plastic dishes, okay? So there'll be a base and there'll be a lid. And it's important that they have nutrients. So there'll be a nutrient agar in the dish that has been specially made to contain sugars, glucose, anything that it needs to grow. So it will need to be able to make amino acids to make into proteins. So if you give it nitrates, it'll be able to do that. And then any minerals it needs as well. We tend to have to go through quite a few stages to grow bacteria on plates, because you'll need to have grown a broth, first of all. You'll need a nutrient broth in a little glass bottle, and there'll be a liquid form of the agar in order to grow like a suspension of bacteria in the first place. And then you use that suspension of bacteria to put onto the bacterial plates for the investigations, to put onto the agar plates. So you have to start somewhere, if that makes sense. You've got to grow them in a bottle first. They're called McCartney bottles. They're just clear little glass bottles, and they'll have like metal lids, and you grow them in a nutrient broth. And once you've given it a few hours and you know you've got enough bacteria growing in there, you can start to plate it onto the agar. In order to do this, you have to keep everything sterile. Everything has to be sterile. You only want to grow the bacteria that you're culturing. You don't want to grow any contaminated bacteria that's got in from the air or from your skin, from any unclean equipment, that sort of thing. Okay? So you only want to grow that strain. Now, on the resource at teachmescience.co.uk for episode 3, there's a resource that has some pictures on it. One of the pictures is how to go about taking bacteria out of a suspension in a bottle and how to put it onto the plate, and the steps we go through to keep sterile. And it's important that you be able to explain those steps, the equipment that we're using, and also how you sterilise all that equipment as you go along. Okay? So first of all, the agar plate that you are using should have been sterilised, as well as the agar that you have made up. So the agar will be made up fresh, it will be sterilised, and then it will be poured onto the plates and left to go hard. The Petri dishes themselves, the plastic dishes, they'll probably already be sterilised. They'll come in a sterilised packet if you buy them. The best way to sterilise things is basically just to heat it using steam at a high pressure, and that should kill any contamination on it at all. All right. What we've got to do now is try and get the bacteria out of the bottle, the suspension bottle of bacteria, and onto the plate without any contamination at all joining it. Okay? So it's important to clean down all the workspace that you've got, so disinfectant all over the area that you're working in, and to light a Bunsen burner, because a Bunsen burner will be quite effective at keeping the air around you circulating, and that way any bacterial cells in the air will quickly be moved away, because like air rises when it's hot, it circulates the air away, so the bacterial cells should circulate away and be away from where you're working.
Fascinating, actually.
It's good, isn't it? In the diagram that we've got at teachmescience.co.uk, you can see a Bunsen burner, a little hand, and it's holding what we call an inoculating loop. So it's basically just
looks like a stick.
It's basically just a metal stick, and it's got a little loop at the end of it of metal. That has to be sterilised before you use it. And the best way to sterilise that is to literally just to put it into the Bunsen burner flame till it turns red, and then take it out and let it cool down a little bit. And that should have completely killed off any bacterial cells that were on that in the first place. We tend to use the loops for streaking. So that means you're going to dip it into the McCartney bottle of bacterial broth, and then you're going to streak across the plate a little bit. We also can spread bacteria, and that's probably the best way to do it, really. Spreading the bacteria is better than streaking. But if you want to spread it, then you need to take quite a large amount. So not just like a inoculating loop worth, but like a centimetre cubed in a sterilised pipette. Put that directly on the surface of the agar, and then you'd use a glass spreader, and the glass spreader would need to be sterilised. So what we would normally do with that is dip it into ethanol, then put that through the flame so it burns off, and then allow it to cool for a little bit, and that will have killed any bacterial contamination on it. So there's two things you can do. You can streak it with a loop onto the surface of the agar plate, or you can spread it about so it goes all over the whole thing. So either one of those really, whether you're streaking directly onto the surface or you're spreading, don't really take the lid off the agar plate for very long. Only lift it a very little bit, quickly do your streak, or quickly do your spread, and then place it back down again. Don't leave the lid off. Don't leave the lid off and just leave it somewhere, because somebody else might pick it up and might use that instead. Always leave the lid on unless you are putting something directly onto the agar surface, because otherwise it may get contaminated with bacteria from the air. Yeah, so we've talked through quite a few bits really. The only other thing I just want to say about the actual taking out of the bottle itself, so if you're taking it from the bottle to put it onto the agar surface, you have to be quite sterile in your action. So you were talking about using your little finger to take the lid off.
Yeah.
That is something that you do, because if you hold the bottle in one hand, use your little finger of the other hand to twist the lid off, you will then be able to keep the lid.
Hold of it and don't have to
That's
put it
right.
down.
You keep the lid in your little finger, exactly like you said then. You don't have to put it down, because if you put it down,
It might get bacteria on it.
it might get contaminated with bacteria. Absolutely, spot on. So you take the lid off, and then you flame the lid in the Bunsen burner. Then you dip in whatever
You
inoculating
don't flame
lube.
the lid.
No, you don't flame the lid, you flame the neck
You flame the,
of the
neck
bottle.
of the bottle. Okay.
What did I say?
The lid.
Oh, did I? I'm sorry, apologise. You flame the lid, like the actual neck of the bottle that you're about to put the pipette or the inoculating loop into. You take out whatever you want. If it's a pipette worth, then you just take like 1 centimetre cubed or something, or a half a centimetre cubed. If it's an inoculating loop, you just dip it in and take it out. You flame the neck of the bottle again, and then you twist the lid back on, and then you can place the bottle down and do your streak or your spread or whatever you're doing on your agar plate. And each of those things that I've talked about are actually in the picture. So if you follow along with the picture from the website,
Yeah, it's all in the diagram.
It's in the diagram, or it'll be in a textbook or something like that. There'll be a picture for you to use. Once you've actually inoculated the plate with bacteria, you're going to need to grow it. So you place the lid on. The lid should be placed on, and it should be taped. So that's so that the lid doesn't come off. And the best thing to do is to just put a little piece of tape round one side and a little bit of tape round the other side, just to keep the lid on. You don't want to seal the lid completely, because then oxygen can't get in. And the bacteria have got all the nutrients they need in the agar, but they're also going to need oxygen to get into the plate so they can respire and grow and replicate and everything. So you do secure the lid with tape, but you don't completely seal the tape off. And that can be a question that you can get in an exam. Why don't you seal it off? And it's because you need oxygen to get in, okay? You need to label the plate with the date that you grew it, maybe your name, maybe the strain of bacteria that you're growing, okay? Make sure it's labelled. Yeah.
What do we grow at school?
We tend to
specific strain.
we can use E. coli. We've sometimes used, I think it's called Micro-Coccus luteus is another strain that we use, because it grows yellow colonies and they're quite easy to see. E. coli tends to grow with little white colonies, and that's okay as well. But remember, they're non-pathogenic. So if you were to get them on your skin accidentally and something was to happen, they shouldn't cause disease. So loosely sealed, but oxygen still be getting in. Don't ever label the lid, always label the base. Because, again, if the lid comes off and gets mixed up with someone else's, then there could be an issue. And with a class of 30 doing the same thing, that is absolutely possible if you're not following the instructions. So keep the lid on your agar unless you are doing something into the agar. That's now going to be incubated for about maybe 2-3 days, which means it's going to go into a warm oven, basically. And we tend to store them upside down so that the agar is at the top, so that condensation doesn't form and drop down onto it. So if you were to store it with the agar at the bottom and the lid on top, condensation will form on the lid, on the inside, and it will drip down onto your agar. You don't want that to happen. So we store it upside down so that condensation can't form and then drip down onto it. And then this is a really important part as well, the temperature that you grow these bacteria at. Why would we not grow the bacteria at 37 degrees?
Is it something to do with that being skin temperature?
It's to do with the fact that that's the body temperature, yeah.
In case it got opened and got on your skin and grew on it?
Well, this is the problem. If some contamination has got onto the plate and there's bacteria from your body on the plate with the strain...
Any of the bacteria that's come from your skin would die if it wasn't kept at 30, if it was hotter than your skin temperature?
No. Say it got contaminated and then you grow it at 37. The bacteria will love that. That's the temperature that it likes to grow at in your body.
The contaminated.
The contaminated bacteria.
So it
the
would grow a lot.
So it would grow a lot. So you could be potentially growing pathogenic bacteria.
Okay.
Which you don't want, which means it would cause disease. So we only grow in school at 25 because it's enough to show colonies growing. So like little circles of bacterial colonies will start to grow on the surface. So it's enough to see colonies on a plate of the strain you're trying to grow, but not hot enough to grow anything that could cause a disease or a problem if you were exposed to it. Does that make sense?
Yeah.
And that is a key question as well in the exam. Why do we grow at 25 and not any hotter?
Mm-hmm. Would they ask you a question like, why do we put it upside down?
Yeah.
Okay.
Yeah. So this sort of thing, as I said, would require practical. Being able to grow the colonies aseptically, which means with no contamination in, is important because the investigation itself would then go on to see if like certain detergents or antiseptics or things like that, see how effective they were. Once you've spread your bacteria on the plate, the most probable next step that you will take before you incubate it is to just take some little paper discs and to dip them in different antibacterial things or detergents or antiseptics and then place them on the surface. Because the second diagram that you've got is an example of what you would see if you were testing the effectiveness of different antibacterial substances and how effective they were at killing bacteria.
Yes.
So just little paper discs, sterile paper discs that are dipped in those and placed on the surface.
About a year ago or so, I watched a video about the effectiveness of hand sanitizer compared to washing your hands.
Right.
And it's a similar sort of thing to this. Like they got the agar and then somebody washed their hands, at 20 seconds in the hot water and they put their hand on. And then another person just hand sanitised and they put their hand on, and it was to see how effective hand sanitizer was. And some hand sanitizer is not effective at all.
No, it's still like loads of bacteria or pathogens and stuff on your hands, isn't there? Yeah.
Yeah. But it's really interesting.
You've got to use specific ones, haven't you? It's like during the pandemic, it was 70% or nothing, really, because what's the point if you don't use that?
Yeah.
What you'll see after the 3 or 4 days of the incubation, if the paper disc, contains an effective antibacterial substance, then you should see what we call a zone of inhibition around the paper disc. Do you know what that means, or do you know what that would be?
My guess is that there's no bacteria because it died or got killed or couldn't grow there.
Absolutely, yeah. So the antibacterial stuff that you put on the paper disc and then placed on the surface has actually killed all the bacteria or prevented the bacteria from growing in the immediate area around, yeah? Mm-hmm. So the bigger the zone of inhibition, the
better the antibacterial whatever you
Exactly, well done. And you can actually then carry out some calculations, like you could measure the area, you can measure the diameter of the zone, work out the area and do some calculations there in terms of that. So a little bit of maths to calculate area of the circle. It's really quite good, isn't it? It's an interesting practical. It's one that I like to do. It's one that I like to teach because being aseptic feels really scientific. When we did it in sixth form, we were very, we wore lab coats and gloves and we made our own agar and we learnt aseptic technique and we grew the bacteria. We made different types of agar and
you got some pictures from
we practiced all the different ways of inoculating plates, streaks, spreads, loads of different things to really get that skill. And that really is a lot of the new information that we needed. So it's just basically talking through how we can grow bacteria on plates and how we stay sterile, and then what we can do with the results afterwards. So should we do some questions?
Yeah.
Okay.
I'm going to ace these questions.
Okay, so there is a resource for the questions because it involves a couple of diagrams. So I'm going to ask Sarah some questions based on these pictures.
Make sure you pull up the resource if you're going to do the questions.
Yeah. Name apparatus A and B.
Oh, so A is the stick. I cannot remember what the stick is called, and that means I'm not going to get my mark.
What's B?
B is the agar plate.
Yeah, so B is the agar plate or Petri dish.
What is A?
Begins with an I.
I'm never going to remember.
An inoculating loop.
Inoculating loop.
Okay.
You better remember that one, guys.
So A is an inoculating loop or an inoculating wire. So that was 1A. 1B. Why should A and B be sterilised before they're used?
So there's no bacterial contamination.
Exactly. To kill unwanted bacteria or microorganisms or microbes. Well done. How should A be sterilised?
A should be sterilised by putting it into a Bunsen burner until it turns red hot, and then you take it out, leave it to cool down for a few minutes.
Absolutely. And then D. Adhesive tape is used to secure the lid of B. Give one reason for this.
To make sure the lid doesn't come off, and to allow oxygen in. To make sure the lid doesn't come off.
Yeah. So it's to allow oxygen in, and then to make sure, it's to make sure that there no bacteria can escape.
What slightly
but
confused
also that
me
none
here
can get in from the air.
was because you didn't say like should be applied to one side or something. It just said should be applied. It didn't. I'm thinking, do I still say the oxygen things? It didn't mention how much tape or anything.
Right. Yeah. So it's
That's
just
a bit
basically
confusing.
to keep the lid on so that no bacteria can get out, no bacteria can get in, oxygen can get in.
Yeah.
Is that okay?
Yeah.
And then E, what is the maximum temperature that you should use in school
25.
to grow bacteria? Yes, well done. Right, question 2 is a bit of a preamble to this, and it's using the second picture, the one with the agar and the zone of inhibitions, okay? This is a bit of a whopper, this is a 6 marker. Okay, but let me read out all the preamble first. A student is given a tube containing a liquid nutrient medium. The medium contains one type of bacterium. The student is told to grow some of the bacteria on agar jelly in a petri dish. Describe how the student would prepare an uncontaminated culture of the bacteria in the petri dish, and you should explain the reasons for each of the steps that you describe.
Okay,
so before you put the bacteria on the plate, what are the steps you've got to take? So pre-inoculation.
Yeah, so how far are we going? Are we saying we have to put on gloves? We have to...
No.
Okay, so we are taking what's it called, that stick again?
Inoculating loop.
Do we start with the inoculating loop?
Yes.
So the inoculating loop goes on the Bunsen burner and gets sterilised.
So that needs to be sterilised in a flame. Well done. Why do we do that?
So there's no bacteria on it.
So there's no bacteria on it. Well done.
Now, do we have to describe that we're taking the lid off with our little finger?
No.
So we have to get the sample tube?
Yeah, so you want to sterilise the loop and then place that into the medium to get a sample of bacteria. Then what do you do with that?
Do we say that we have to sterilise the rim of the tube or anything like that?
I don't think you need that much detail, no.
Okay. If I'm thinking like I want to go full detail.
Yeah.
Then we're going to take the lid off the agar.
Take the lid off the agar as little as possible.
Put it on.
Streak it on.
It on. Put the lid straight back on.
Put the lid straight back on.
Then we want to sanitise or sterilise the loop again.
Yep.
Put a bit of tape on it.
Put a bit of tape on it. Why?
The lid doesn't fall off. No, nothing gets in or out.
Yeah.
Apart from oxygen.
Yeah.
And then we're going to write our name on the bottom or something.
Yeah. So label it and then
put it into the thing for 25 degrees.
So 25 degrees incubation for 3 days, 3 to 4 days.
Mm-hmm.
Is that okay?
Yeah.
Right. After the culture has been prepared, the student added 1 drop of 5 disinfectants, that's A to E, onto the culture. The diagram shows the appearance of the petri dish 3 days later. So you can see in the picture, each one's labelled and each one's got a different sized zone of inhibition, okay? These are areas on the agar jelly where no bacteria are growing. Why?
Because the disinfectant is there.
Yeah.
And the disinfectant isn't allowing any of the bacteria to grow.
Okay, so it's either not allowing it to grow, so it's preventing its growth, or it's killed it. Yeah, well done. The student concluded that disinfectant D would be the best for using around the home.
Yeah.
Give one reason why the student might be correct, but also one reason why the student might not be correct using that around the home.
It might be too strong.
Right, exactly. So one of the reasons why it might not be correct are that it might actually be harmful to people or your pets or,
yeah,
you know.
It might be harmful, but it's good because it looks like it isn't letting any bacteria grow. It's the strongest of all the disinfectants.
How would you describe that, though, on the picture? Like, from the diagram, how would you describe it?
It's got the biggest ring of death.
It's got the biggest zone of inhibition.
Zone of
Ring
inhibition.
of death. It's got the biggest zone of inhibition, or it's got the largest area where there's no bacteria growing. Yeah, good. So that's really the only reason why we would say that, because that's all we can see.
Yeah.
But there could be lots of reasons why he's not right, and like you said, it might be too strong. It might not work with all bacteria.
Can you use that my word in there? It might be too strong. Would that get you a mark?
Yes.
Okay. Good.
Yeah. You might need different concentrations. So it might be like the strong concentration might be too strong.
Water
It might need to be weaker, or it might not last very long. Yeah, that's pretty much sort of the exam questions there.
The C is a rubbish one.
C doesn't allow anything to. Yeah, I mean it might just be that particular strain it doesn't work on, but yeah, still. Shall I do the roundup?
Yeah, go ahead.
Binary fission is how prokaryotic or bacteria cells divide. This can happen up to every 20 minutes, and a copy of the chromosome and the plasmids within the cell are made, and then after a little while the cytoplasm will start to divide and the cell membrane will start to pinch, so it separates into two new cells. Each cell will have one chromosome of its own and some of the plasmids. In order to investigate non-pathogenic microorganisms in school, bacteria can be grown on a petri dish with a nutrient agar. The nutrient agar must contain the glucose and sugars, carbs, nitrates and different minerals for the bacteria to grow. When growing bacteria, you have to keep everything sterile so that only the strain you want growing is there and no contamination. Agar is made up fresh and sterilised. The sterilisation is usually done in an autoclave, heating at a high pressure with steam, and that will kill any contamination in the agar or on the petri dishes. This can then be poured warm and allowed to set. The bacteria that you want to inoculate these plates with is grown in a separate bottle in a nutrient broth. This can be streaked or poured and spread onto the plate. It is important to have a Bunsen burner lit at all times near your work surface to clear the air around you where you are working of any microorganisms. For streaking bacteria, you have to use an inoculating loop. This is a small metal wire loop and must be sterilised by putting it through a flame until it is red hot and then allowing it to cool. You can then dip it into the nutrient broth and streak the agar. When you open the bottle, flame the neck before you close it again. When you streak the agar plate, only lift the lid enough to put the loop in and to streak. For spreading, use a glass spreader. Dip that spreader into the ethanol, flame it and allow it to cool. Must use a sterilised pipette to draw up a specific amount from the nutrient broth, and then pipette that onto the surface of the agar, and use the spreader to spread it across the surface. All these steps will ensure all equipment is sterile, so that no unwanted bacteria are on it, and no bacteria from the air can get onto the plate. To grow the bacteria, the plates need to be sealed loosely with tape, enough so that oxygen can get in, but not completely sealed. Store in an incubator upside down to prevent condensation falling on the surface. The incubation must be at 25 degrees so that no pathogenic bacteria will grow. Typical investigations involve testing different antibacterial substances or antiseptics or detergents to see which is the most effective. You must use small sterile paper discs soaked in these substances, and they can be placed onto the surface of the spread bacteria before it is incubated. The more effective they are, the more bacteria around the disc will have been prevented from growing. This is called a zone of inhibition. Calculations can be done to calculate the biggest zone of inhibition and therefore the most effective at preventing bacterial growth. What are got any takeaways?
Sterilising is really important to prevent bacteria. What was the word you'd say?
Any unwanted bacteria and any contamination.
Contamination is the word. Prevent contamination. You sterilise things by putting them in a Bunsen burner or in ethanol. We put the agar plate upside down when it's heated, so any condensation doesn't fall onto the agar.
Well done. Yeah, good.
Is that the bell?
Yeah, that was well.
Oh, great. I've got PE.
Okay, what PE?
I forgot my kit. Can you write me
You're
a
going to
note?
have to wear some disgusting stuff out of
Can
the
you
lost
write
and
me a note
found.
say that I've got an
No,
injury? Why not?
No.
Why not?
No.
But I've got... But
No.
fine.
Get to PE. Go on, off you go.
My
See ya.
name's
Bye.
Matthews.
And I'm Ria Corbett.
This has been Teach Me GCSE Biology, and you will hear us next time.
Bye.
Bye.
Bye, 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 bye.
I can't believe you won't write me that note.